this is Dr. B. This is a short video to kind of show you what level of expectation I'm looking for in documentation in OneNote. So basically, let's just grab this, whether you're in Bio 262 or Sci 130, doesn't matter. So let's assume here that this is the mothership and this is what Dr. B posted. And if you look over here, I made myself a student. And so I really have to kind of transfer the idea of what's over here over to my notebook, which by the way would be like your notebook or any of the other people up here. So let's just say the first thing that this is about, gel electrophoresis. Maybe this is something we did this day. And as you know, if you did do something, you would have had other notes in your notebook that you took or whatnot. So what would I do? The first thing I would do is basically, call this a particular page, call it gel electrophoresis, something like that. Helps if I spell it right. And so basically from there, the first thing I do is put on supplies or materials. You could basically say it either one. And what, so now I'm just being a student. But now if I look back, well, what are the supplies? Well, we obviously used our DNA samples from PCR. And we also used, what else did we use? Oh, look, well, we use this. All right, Agros BP 160-100. We use that and the lot number because I want to make sure I have the right one. BP1, the lot number on that was 218051. Great. And then the next thing we used was the gel box. And so again, I'm not going to write it, but you would then do that. Biorad, or I'll do it. Why not? Mini subcell GT. And is there any sort of like, serial number or anything like that? Let's go back and look and see if there is one. There is 329 BR03. And there must be more to that. 5334. Or actually, it's hard to see. So, yep, 5334. See how I can make it bigger? Great. Was there anything else that we used in that lab? The DNA gel stain. Again, I can make this bigger if I need to just kind of see what it says. DNA gel stain. So you can kind of see what I'm doing. DNA gel stain. And that one would be 236, 1836. Right. So that's basically everything for supplies, and I could either do it there and continue, or I can go down here and say procedure, and this is from what I had on the board for whatever class or what you'd written in your notebook. First thing would be like make a 1% agarose gel. Actually, if I change it, make 50 milliliters of a 1% agarose gel by adding 0.5 grams of agarose to 50 milliliters of 1x SB buffer. That's enough. I don't need to go, and obviously it's understood that I microwave and whatnot, but after that I also want to make sure I add 5 microliters of, I can just say now basically DNA gel stain because it knows what it is above. And so basically that's what I did. And then from that part you could maybe, well, what'd you do after that? Basically you could load, all right, for clean samples, and those of you in class, you know what that means, add 3 microliters of loading dye to 15 microliters of DNA in a tube and mix. See, I'm kind of writing it very short. Then it would be load 15 microliters of clean DNA, right? And 15 microliters of green DNA. You guys know what that means. Again, if you're inside 130, you have no idea what I'm talking about. That's right. That's no problem though. Into each labeled well. What I'm then going to do is show you what I could do from there. I'm going to use Portia. Portia already kind of went ahead here and had all of her stuff in order and basically did this today. So you can see that Portia not only went into way more detail than I did, and that's totally fine. I wanted to show you the minimum. But you can see here that all the lanes are figured out. Voltage 290 for 20 minutes. What's in each lane? She already pulled up a picture. She took a picture off the screen of the gel. So this is what I would expect. Something like this is really, really good. Just I should be able to follow what you did. And so that's the idea of me posting stuff here and you being able to put it onto your particular page here. Also, you don't have to do it this way. You could also do something like what Lily does. And Lily likes to just write only, and I think Brooklyn too, I don't know if she's not there yet, but Lily likes to just write in her lab notebook and that's it. And that's perfectly fine. You can see that at the end of every day, what Lily does is just posts a picture of what was in her lab book. It doesn't matter which one you do. And I can make that bigger if I need to. It just helps that you document. The whole point of this is to be able to follow what you did, so that we can go back if something doesn't work. And so hopefully that little thing I did here gives you some kind of help in determining what you would be doing to help with some of the actual work for the loading of the OneNote system, which I like to call it. So there you go. And if you have any questions, let me know.